Hello everyone, welcome to Study with Manjula and I am your friend Manjula Sri. In this video we are going to start a new topic, that is structure of chromosome, cell cycle and cell division. It is of class 9 ICSE syllabus. Before going to that, we have to clear some of the concepts about what is a cell. Let us go. Here I have taken a cell. I'll draw a cell here. Right, inside cell what we will have? A nucleus, right? A nucleus and the organelles. Now I have taken out this nucleus and I'm drawing it a bit larger. Inside the nucleus, which parts we will have? A nucleolus we will have, right? A nucleolus, and the most important thing we will have is the chromatin fiber. What? Chromatin fiber. We are clear about the cell, what is inside the cell and what is inside the nucleus. Now why we are learning, why we need to learn about this nucleus and this chromatin? Let us see what is a heading telling. Where it went? Right, structure of chromosome. Before going to that, what is a chromosome, I just want to clear the concept what is cell cycle and cell division. First, before learning the cell cycle and cell division, you should know what is a chromosome and its structure, where it is present. Now what is a cell cycle? It is the cyclic, with the word you can understand that it is related to cell. So the cell cycle is nothing but the life process of the cell, and the cell division is if the cell divides, then only the human body grows, right? I just want to clear one concept. See, if there is a child of 1 year, you have seen a child of 1 year, he is a baby, he will be a small baby, right? After 5 years, you will see the same child. If you see the same child after 5 years, many changes takes place in him. His hair grows, right? He will have eyes, nose, and he will become taller, right? He will become taller, stronger. He will become stronger, and he will also attain the characteristics of his parents even. Yes or no? Some children looks like their father, or some children looks like their mother. How is it possible? How they are growing? For knowing everything, you should know what happens in our body. You know, cell is the basic structural and functional unit of all living organisms. If you know about the basic structure, that cell, what's happening inside it for the growth of a human, if you will know the cycle, the life process of that cell, you will understand what is happening in our body while we are growing. Understand? Now, the part which initiates the cell cycle, the most important part is nothing but the chromosome. What it is? Chromosome. Now what is this chromosome? I have drawn a nucleus here, right? I have drawn a nucleus. Inside that I have said there is a chromatin fiber. This chromatin fiber, after a few days, it just get condensed. Condensing means it will become thick. What it will become? It will become thick. It will become thick. And after few days, that same chromatin fiber, it will become chromosome. It will become chromosome. Now what is that chromosome? We will learn it. Chromosome was discovered by a scientist called Walter Fleming. Walter Fleming. Walter Fleming was a German scientist. He have noticed these chromosomes inside rapidly dividing cells. He have noticed these chromosomes inside an amphibian known as salamander. It is an amphibian. A salamander is an amphibian. Amphibian means an animal which can live both in water and on land. Who is the person who have discovered chromosome? He is Walter Fleming. Okay, note down this point. It is a very important big point you will get in the exams as an MCQ points or any one word answers. Right? Now let us learn how this chromosome is formed. Now the chromatin fiber which gets condensed, it will form a chromosome. How? See, this is a chromatin fiber, right? This chromatin fiber, what happens is, it will convert into chromatids. What? Chromatids. Like two arms it will become. It will become like, sorry where it went. See, it will just become like two arms. These two arms are called chromatids. What? Chromatids. Chromatids. It will become what? Chromatids. These two arms you are seeing, these two are called chromatids. Right. So this chromatid, after few days, it will duplicate itself. It will duplicate itself. That means it will become double. It will become double. This structure is nothing but, it's called chromosome. What it is? Chromosome. The duplicated chromatid is known as chromosome. This I am giving a numbering. This is 1, 2, 3 and 4. There are four arms. What? Arms of chromosome, which is nothing but chromatids. What? Chromatids. I have said these arms are called chromatids. Now these chromosomes have 4 arms, which are the chromatids. Okay, and here the central part you can see. That central part is nothing but centromere. What it is? Centromere. The centromere is the point of intersection between the chromatids or the two arms of chromosome, you can say. Now here you are going to learn the 3 words, that is, just without getting confusion, I am going to tell those words, that is, they are, one is chromatin fiber, chromatin fiber, the other one is chromatid, and other one is chromosome. These are the 3 words. Now what is the difference between 3 words is, chromatin is the fibrous substance present in the nucleus. Okay, chromatid means the arms of the chromosome. What? Arms, ARMS, arms of chromosome. Chromosome is the highly coiled and condensed, that means the thickened part of chromatin. Chromosome is what? The highly coiled and condensed chromatin fiber. I'll write the definition. See, the highly coiled and condensed chromatin fiber. Chromatin fiber. This is the definition, you can note down. The highly coiled and condensed chromatin fiber is called chromosome. Understood? Okay. Now in the definition we have seen that coiled. What? The word coiled. We have said also condensed. We have said condensed because that chromatin fiber is condensing itself into chromatids, and chromatids are duplicating themselves and forming the chromosome. Now, why the word coiled is used? What structures are coiling themselves, that we should know. Before that, we should know the structure of chromatin. Okay, chromatin, we know that is the fibrous material. It is composed of two substances. It is composed of two substances. One is DNA, and other one is histone. One is DNA and other one is histones. These two composing together, we will call it as a chromatin. Or chromatin is constitutes these two substances, that is DNA and histone. First, we will learn about DNA. Right. What is DNA? Oh, where it's going? Right. DNA. You know what is the full form of DNA? It's deoxyribonucleic acid. Nucleic acid. Deoxyribonucleic acid is the full form of DNA. Right. Deoxyribonucleic acid is the full form of DNA. DNA was first discovered by person, means the first who studied about DNA was Rosalind Franklin. But he was failure. Franklin, but he became failure. He only studied, but he did not discover. He was not able to discover. He just studied the shape of the DNA, but he did not discover. The structure of DNA was completely discovered by two people. That is, they are called Watson and Crick. C-R-I-C-K. Crick. Watson and Crick. These two people completely discovered the structure of DNA, and they proposed the model of DNA, that is the double helical model, they will say, and they got the Nobel Prize in 1962 for discovering the structure of DNA. Okay, now we will see what is this DNA composed of. This DNA is composed of three things. One is phosphate group, other one is nitrogenous base, and third one is sugar molecule. Right. Sugar molecule. Right, three things, that is phosphate, one, and other one is nitrogenous base, and other one is sugar molecule. These are the three things. The phosphate, you know, it is an element, and one sugar molecule, and there are four nitrogenous base. The four nitrogenous bases are, first one is adenine, other one is thymine, and the next one is guanine, 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 and other one is cytosine. These are the four nitrogenous base. Okay, these 4 nitrogenous base, they are just paired with each other. Adenine we can write in short form as A. Thymine you can write as short form T. Guanine you can write as G, and cytosine as C. Okay, I have said that the 2 nitrogenous base combined with, they are only paired with the 2 nitrogenous base. Adenine always paired with thymine, and guanine is always paired with cytosine. Adenine is paired with thymine with 2 hydrogen bonds. Wait, I'll change the color. With 2 hydrogen bonds. With 2 hydrogen bonds. Guanine is combined with cytosine with 3 hydrogen bonds. What? 3 hydrogen bonds. Okay, you just be clear with this concept that there are 4 nitrogenous base, that is adenine, thymine, guanine and cytosine. Adenine always combines with thymine with 2 nitrogen bonds, and guanine always combine means they just attract each other, guanine and cytosine with 3 hydrogen bonds. Understood? Now I'll show the structure of DNA. How is it composed? Right. Now here I have a phosphate group. Here I have a phosphate group. Right. Next I have taken one. Wait, again the same color I got. Yeah, I have taken one sugar molecule and one color fillet. Oh, why it's not coming? It went white. Okay. So this is one sugar molecule and one nitrogenous base I have taken. So these all are combined with one bond, that is known as phosphodiester bond. What? Phosphodiester bond. Phosphodiester bond. Okay, phosphodiester bond. This is also one bit point. Please remember this. The 3 components of DNA are combined with phosphodiester bond. Okay. I'll just rub this and I'll draw the DNA structure. This single structure you're seeing, what you're seeing, this one is, this structure you're seeing, this structure is known as nucleotide. When the series of nucleotides are arranged in a sequence, then that is known as DNA. Now I'll arrange that nucleotide in a sequence. Right. Now I had one phosphate group. Now here I'll have another phosphate group. Here one phosphate group. Here one phosphate group. Next one nitrogen, one sugar molecule. It is a pentagon. One sugar molecule. One sugar molecule. Right. Sugar molecule and nitrogen bases. Nitrogen base. Right. And I will bind these with the them right. Now this one I'll connect like this. I'll just wait. Here I should connect like this. This one I should connect like this. Right. Now this is a DNA molecule. I just have given just a rough diagram. A DNA, it will be like this, and there are many series connected like this. Okay, this is the phosphate, this is the sugar molecule, and this is the nitrogenous base. This is the sugar molecule and this is the nitrogenous base. This is the structure of DNA. This is a scientific structure of DNA. In videos, if you have seen, the structure will be somewhat like this. You will see two strands like this, and here you can find like this. So this is the DNA actually. I have drawn two strands like this. See, here one phosphate group, here one sugar molecule, here nitrogen base, here one sugar molecule, here one phosphate group, and here one nitrogenous base, and here like that I have drawn, right? See, what I have drawn is, this is one strand of DNA and this is one strand of DNA. There are two strands of DNA. These both are anti-parallel to each other. They are anti-parallel to each other. What is anti-parallel? See, if there are two lines at equal distances at every point, then it is a parallel. But if the two are parallel but runs in opposite direction, then that it is known as anti-parallel. See, here the phosphate group is, the sugar molecule, they have the pentose one like this, but here they will have like this. This is flowing downwards and this is going upwards. This is an anti-parallel one. Okay, you can say that the DNA strands are anti-parallel. Till here you have understood what is a DNA. In next video we will see about the histones and the coiling of the chromosomes with the histones and the structure of chromosome in detail. Till then, thanks for watching.